Hi, this is Paul Norman here. I thought I'd tell you a bit about a project I was involved with a couple of years ago with some people from one of the hospitals in Leeds, and it's to do with antibiotic resistance. There's quite a few long medical terms here, which I must confess I don't necessarily understand myself, so we're not going to spend too much time worrying about those. Anyway, Tim Kemp was the guy who got in touch with me, and he was then a registrar dealing with infectious diseases and so on. So he was interested in antibiotic resistance and can we estimate the population level distribution of resistance to a particular antibiotic. I expect you probably know that some bacteria cause infections of the urinary tract or the abdomen and so on, and that eventually, or actually relatively quickly, they can become resistant to the majority of antibiotics. They make an enzyme called ESBL, and that's about the only abbreviation of a long word that I'll continue to use apart from antibiotic resistance. So this is increasing. An ESBL can be carried in the bowel without causing any symptoms, but many people can get serious infections. So this was a pilot study, and Tim approached me to see whether I could do the GIS type work with this. Leeds Teaching Hospital, Bradford Teaching Hospital, and School of Geography provided data and so on. So this was the first map that I produced, and those dot maps there are proportional symbols of the number of people who had stool samples tested in a particular lab, and the larger the dot, the more the resistance. So the class intervals are proportions of tests that were carried out that were found to be resistant to the antibiotic, and in this case the class intervals are chosen for being sort of logical progressions of round numbers, but the highest one there, 0.25 and upwards, 0.25 or a quarter is considered to be the proportion of the population, if they are resistant, that is representative of community resistance to an antibiotic. So that's the kind of warning level that we don't want to see. What you can actually see on those slides is, particularly in the southeast of Bradford, that's around the city centre sort of area, collections of large blobs. Leeds seems to be a little bit more even, sparse, whatever. So in terms of the types of work that we've been doing in this module, our eye might be suggesting that there might be clusters in southeast Bradford of high resistance, and perhaps that isn't the case in Leeds. The first thing I did with this to see if we could estimate where we hadn't got samples, so using inverse distance weighting as an interpolation measure. So that takes the sampled points that have been through the lab and then interpolates those across space. And again, I've used the same class intervals to sort of indicate those locations where there might be higher resistance to the antibiotic. But if we're going to think about spatial clusters where people live together closely in high density, that's going to bias the sort of occurrence of any particular measure. So it will be higher than we would expect where there is high population density. So these maps are the lower superadapted area population densities. So the darker colours are the higher densities. And again, that area of Bradford I was talking about is the highest population densities across the whole of the district, and Leeds, again, not far away from the city centre, that's where there are higher population densities too. And both are typical of many British cities and so on, whereby the inner parts of cities have higher population densities, and towards the external, moving towards more rural areas, are lower densities. So is there any spatial clustering? Here I use the geographical analysis machine, so that's the older software developed originally by Stan Openshaw, and whilst we haven't used it on the module, it is a very straightforward way of showing clusters, I happen to be able to say it's a complete pig to get the GAM outputs to display in ArcGIS, and I haven't even tried it in Arc Pro yet, but it does work well as a clustering algorithm, but perhaps superseded by some others. Anyway, I used it here, and what that does show is a cluster in that southeast portion of Bradford. So higher incidence of resistance to the antibiotic than we would expect by chance, effectively. So to sum up, our work here was only for a small study area. It wasn't a complete coverage of lab tests, it was just from one lab. So on the aggregated postcode level records up to postal sector, and then I used the centroids for the mapping and for the interpolation, etc. So it lacked the detailed resolution that we might really want with here. What we'd like to do is to directly compare with GP prescribing, because that is one of the things that's cited as being leading to community resistance if GPs are prescribing antibiotics more than they probably should. So the future plans, well, this was the future a couple of years ago, was to address the above, i.e. do more of this, and perhaps explore use of more interpolation and spatial clustering techniques than I did there, particularly if we had unit postcode centroid location available. Sadly, we never got funding to do more work than this, although we had some good feedback from the referees of funding proposals we put in. Since then, Tim Kemp has moved on, and he's now a consultant in this kind of work in Stoke-on-Trent, and I do occasionally swap emails with him, and he's a nice chap, and it was lovely doing this particular piece of work. So I hope you can see the type of thing that I did there, the way I stepped through some of the thinking, and that the range of techniques we've used in the module were the type of things that I used here, even if not specifically each time. That's what we do. We kind of pick up on techniques and we try them and use them in particular ways. If anybody wants to know any more about this, then by all means drop me a line and I'll explain more if you wish. Oh yes, forgot about this bit. Great presentation, Paul. This was part of the extra work that we wanted to do. The dots on these maps are in fact unit postcode, so that's the postcode distribution across Leeds and Bradford. And what we actually wanted to do, and this is what the funding proposal was for, was to set up kind of a live reporting system so that if somebody visited the GP and they had a urine infection or something, the GP could put their postcode into a database, and it would tell them, the GP, whether that person lived in a location where we were estimating there was already resistance to the commonly prescribed antibiotic, and then the GP could not prescribe that one but prescribe something else. Again, that was part of the funding proposal, and we thought it was a great idea, as did other people, but as I said before, sadly we didn't get the funding. And that really is the end. So there's just one reference there from something on the first slide which explained some of the technicalities of the biochemistry, which of course I don't understand, and say thank you very much for the funding we did get, which was just a few £100 to be perfectly honest, but any money is money, so thanks very much. Anyway, I hope you found that interesting. Shout if you want to know more. Cheers.